In Chapter 14, we looked at some of the exceptions to the rule in patterns of Mendelian genetics. So, for example, with epistasis, instead of seeing the expected 9:3 to 3:1 pattern of phenotypes for genes that assort independently, we saw a 9:3:4 ratio because one gene masked the effects of the other. But the laws of independent assortment and the law of segregation still applied in all of those cases. Now we're going to talk about some true exceptions to the rules that don't even follow the laws of segregation or the laws of independent assortment in the way that other genes do. So some examples that we're going to talk about are genomic imprinting and genes that are stored within other places than the nucleus. So what other organelles have DNA, have their own DNA? So for animals, we have mitochondria, and then for plants, they have both mitochondrial and chloroplast DNA that can lead to different patterns of inheritance. Your mitochondria came from your mom, and we can trace maternal lineages through mitochondrial DNA. Your paternal lineage can be traced through the Y chromosome if you're a guy, and then these extra nuclear genes in the mitochondria and the chloroplast can be actually passed out kind of mismatched and mixed ways. So not every descendant gets the same mitochondria or chloroplast with the same genes. It's kind of random on how the cytoplasm is divided up. So one specific example of a true exception to the rule that we're going to talk about are genes that are imprinted. Imprinting has to do with epigenetics. Epigenetics means above genetics or beyond genetics. So they're heritable changes that are not caused by changes in the DNA sequence, not changes in mutations. Okay? So just a basic example of this, the fact that all of your cells have the same DNA, but your skin cells are skin cells and they don't randomly turn into muscle cells at any point, has to do with epigenetic changes, which we'll talk about more in Unit 4. The specific example that we're going to talk about today is genomic imprinting. And we're going to give one example of X inactivation and then one example of autosomally imprinted genes. So it's been known for a long time that in female mammals there's this dark staining spot. It was first described by Dr. Barr, so it's called the Barr body. It shows up in females, in mammals, but not in male mammals or other organisms. And that dark staining spot, you can see here, it's a little darker blue than the rest of the DNA in the nucleus, also stands for markers that indicate that it's genetically inactive, that it's off. Okay. So in the 1960s, Mary Lyon had a hypothesis about what this Barr body was. And she had a couple pieces of information that she used to formulate her hypothesis. She knew that in mice that had only one X chromosome, they were females and they were normal, and in mice they were even fertile. In humans, XO females are also pretty normal and healthy, but they have some infertility. Other things that she was aware of is that sex-linked mutations in females tend to have a mosaic pattern, meaning it's blotchy, it's splotchy. So tortoiseshell cats or calico cats have blotches of fur in different colors that are not found in male cats. Okay. And then another piece of information she had was that this dark staining Barr body was seen in every mammal that had been looked at, from cats and humans and monkeys and rats to even possums. So Mary Lyon proposed that what that dark staining spot was the X chromosome. And it was just one of the X chromosomes that was genetically inactive or shut down. And she also proposed that which X chromosome was shut off was random. So it could be the one that the female got from its mom or from its dad. So a lot of research went forward after this point to validate her hypothesis about X inactivation. So it's true that in female mammals we have two X chromosomes. Okay? And early in development, one of them expresses a non-coding RNA called exist. And what exist does is it functions as an RNA. It doesn't code for a protein. It's non-coding. And that exist RNA goes back and it coats the X chromosome that first turned that gene on. And that coating of RNA attracts other proteins that cause this X to shrivel up and compact down into a tiny little bar body. If a female is heterozygous, meaning she has the dominant copy for one gene on one X chromosome and the recessive on the other, if X inactivation happens on this X, in those cells she'll display the dominant phenotype. But if just by chance it was the chromosome that had the dominant allele that was shut off, in those cells she'll have the recessive phenotype. So for female mammals, a particular cell is actually only expressing one X chromosome at a time. Actually a counting mechanism that makes sure that one X and only one X gets turned off. So it's random which X gets turned off, but whichever one expresses exist first gets shut off in the normal female. In individuals that are XO, that have just a single X chromosome, no exist is expressed, and so the single X chromosome is expressed. If you have an aneuploidy with triple X, okay, there's a counting mechanism, and all but one X chromosome gets shut off. In platypus, which have like 6 pairs of sex chromosomes, all except for one X chromosome gets shut off by the same mechanism. So the purpose of X inactivation is to do dosage compensation, to make sure that females who have 2 X chromosomes express the same amount of genes as a normal male. So remember, males just have one X chromosome. The Y chromosome has totally different genes on it. So dosage compensation by X inactivation makes sure that dosage, or the amount of genes that are expressed from the X chromosome, matches males versus females. Now, probably this X inactivation is actually slightly leaky. There are some genes that escape X inactivation and are still expressed, which lead to some subtle differences in males versus females. Interestingly, I think other organisms do dosage compensation in different ways. In fruit flies, for example, females are XX and males are XY. But in fruit flies, what they do is they double the amount of gene expression from the X chromosome in the males so they catch up to the females. In other organisms like C. elegans, the females have 2 sex chromosomes and males have only 1. And in C. elegans, both X chromosomes reduce gene expression by half to do dosage compensation. So X inactivation breaks the rules of Mendelian genetics, because even though a heterozygous female has 2 copies of the gene, she only expresses one or the other in a particular cell at a particular time. So really, if we look at an entire human being, okay, she might be expressing the dominant allele in this patch of skin and the recessive allele in this patch of skin. And that's exactly what we see in a calico cat or a tortoiseshell cat, where in some splotches the dominant allele is expressed and in others the recessive allele is expressed. Now this can be confusing if we go back and think about our X-linked gene Punnett squares, but because of the timing of X inactivation, the splotches of one or the other are usually evenly dispersed. So in a female who's heterozygous for red-green color blindness, a particular eye cell might actually be red-green colorblind, but the one next to it is normal color vision, and so overall she has a normal phenotype. So if you're working with genetics problems asking about X-linked genes, you can forget about what you know about X inactivation. What you should know about X inactivation is that it only happens in female mammals, and it's random which of the 2 X chromosomes gets shut off. And the mechanism for shutting them off is a non-coding RNA called exist, which silences one of the X chromosomes. Every cell in a female's body, one of the X chromosomes is randomly shut off. But those marks that silence that are actually erased and reset during meiosis or prior to meiosis in the germ cells so that females pass functional expressing X chromosomes to all of their offspring. So this is called imprinting, and we see it not just on X chromosomes, but also on autosomal genes. So we're switching from X chromosomes to genes that are on autosomes, not the sex chromosomes. Genomic imprinting has to do with whether a gene is expressed depending on who you inherited it from. Mendelian genetics tells us that you have 2 copies of every chromosome, one that you got from your mom and one that you got from your dad. And so if you're heterozygous and you have both a dominant allele and a recessive allele, you'll have the dominant phenotype. But in genomic imprinting, certain genes get shut off based on the parent they were inherited from. So for some genomically imprinted genes, whatever copy of that gene you got from your dad, it's shut off. And so if you were heterozygous here, but the dominant allele was the one you got from your dad, even though you're heterozygous, you would only express the allele that you got from your mom. Other loci are imprinted differently. So at a different locus, you could still get dominant from your dad and recessive from your mom, but at this locus, it's imprinted maternally, meaning whatever copy of the gene you got from your mom, that's the one that's shut off. And then in that case, you would display the dominant trait. The way this was discovered was studying the IGF-2 locus in mice. Researchers were trying to study the IGF-2 locus, which is the insulin growth factor 2, and they were just trying to set up crosses. So they started with true breeding homozygous dominant parents and true breeding homozygous recessive parents. But they saw, depending on how they set up the cross, they got different things. If mom was homozygous recessive and dad was homozygous dominant, all the F1s were heterozygous and all of them were normal size, which is what you would expect. But what they noticed is that if mom was homozygous dominant and dad was homozygous recessive, all the offspring were heterozygous, yet even though they were heterozygous, they were tiny. They had the growth factor mutation phenotype, even though they were heterozygous. What they were able to figure out is that the IGF-2 locus is maternally imprinted. And what that means is whatever copy of the gene you get from your mom is actually shut off. And so even though these guys were heterozygous, they were only expressing the dominant allele from their dad, and that's the phenotype they had. But when it was inherited this way, the copy that they got from their mom was shut off, and that was the dominant allele, so they had the recessive phenotype. Interestingly, there's another locus called the H19 locus, which is imprinted the other way, meaning the copy of the gene from dad is shut off, and so the phenotypes flip. So for imprinted genes, even though you have 2 copies of the gene, you only express one or the other, not both at the same time. And imprinting only happens in mammals, and it has to do with differences in evolutionary interests between moms and dads. Moms want to make sure that their babies get a fair chance, that all of their offspring can get big, so they reduce resources, whereas dads want to make sure their offspring are bigger, and so they try to grab more resources. The thing that you need to know is that even though there's 2 copies of the gene, one of them is shut off. Genomically imprinted loci are on autosomes, so it doesn't matter whether the offspring are male or female, but what determines whether the chromosome is shut off is the sex of the parent that they inherited them from. And then these F1s, when those germline stem cells go through meiosis, whatever imprints are in here will be erased, and then what's imprinted in the next generation will be determined by the sex of the individual that's passing the genes to the next generation.